Swayam Prabha. Digital India. Educated India.
When inbreeding occurs, the frequencies of genotypes change like this. With inbreeding, the proportion of heterozygotes decrease by a factor 2 FPQ, and half of this value, that is FPQ, is being added to the proportion of each homozygote. There is something called inbreeding depression as well. Let us assume the frequency of a recessive allele that causes a genetic disease is 0.01. If the population mates randomly, that means if the F value, that is inbreeding coefficient, is 0, the frequency of individuals affected with disease, that is small a, will be q square. So square of 0.01, that is 0.0001. So only 1 in 10,000 individuals will have the disease. However, if F equals to 0.25, then the expected frequency of the homozygote genotype is q square plus 2pqf, as we just discussed in the previous slide, and this value comes out to be 0.0026. Thus, the genetic disease is 26 times as frequent at this level of inbreeding. So you can just see, if the inbreeding changes from 0.0 to 0.25 in terms of inbreeding coefficient, this disease has become 26 times as frequent as in the condition where inbreeding is not there. This increased appearance of lethal and deleterious traits with inbreeding is called as inbreeding depression. The more intense the inbreeding is, the more severe the inbreeding depression is. Now let us move ahead towards various evolutionary forces which can upset the Hardy-Weinberg equilibrium. And as we just mentioned in the previous slide, these can be mutation, migration, genetic drift, and natural selection. So let us take them one by one and see how they change the allelic frequencies. Mutation as an evolutionary force to change the allele frequency of a population or to upset the Hardy-Weinberg equilibrium. Mutation can influence the rate at which one genetic variant increases at the expense of others. Now let us try to understand it mathematically. Consider a single locus in a population of 25 diploid individuals. So the number of alleles will be multiplied by 2, that is 50. Now let us assume that there are two different alleles, G1 and G2, whose frequencies are depicted by P and Q respectively. If there are 45 copies of G1 and 5 copies of G2 initially in the population, so P is 0.9 and Q is 0.1 respectively. Now suppose that a mutation changes a G1 allele into a G2 allele. After this mutation, there will be 44 copies of G1 instead of 45 and 6 copies of G2 instead of 5. And the frequency of G2 will increase from 0.1 to 0.12. So that means one mutation has changed the allelic frequency. Now this is the case of forward mutation, that is from G1 to G2. The change in G2 as a result of mutation equals the mutation rate times the allelic frequency. So the change in Q will be mu, that is forward mutation rate, multiplied by the P, that is allelic frequencies of allele G1. Now there is a possibility that reverse mutation also occurs, that is from G2 to G1. So this change in allele frequency of Q as a total of forward and reverse mutation becomes mu P minus mu Q, where delta Q represents the overall change in the allele frequencies, mu represents forward mutation rate, mu represents reverse mutation rates, P and Q respectively shows frequency of allele G1 and G2. If the only evolutionary force on population is mutation. That means there is no other force, but only mutation is there. The allele frequency change with time that we have just seen. Eventually, it reaches equilibrium and is determined by the forward and reverse mutation rates. The effect of typical mutation rates on Hardy-Weinberg equilibrium is negligible, and many generations are required for a population to reach mutational equilibrium. But remember, we are talking about this when there is only mutation as an evolutionary force and there is no other force. Moving ahead to discuss about migration as an evolutionary force to change the allele frequency of population or to upset the Hardy-Weinberg equilibrium. Assume the frequency of an allele in population 1 as Q1 and in population 2 it is Q2, and in each generation a representative sample of the individual in population 1 migrates to population 2 and reproduces there, adding its genes to the total gene pool of population 2. So the situation is clear. There are two populations, population 1 and population 2. The frequency of particular allele, that is Q, is Q1 in population 1 and Q2 in population 2. Some representative members of first population migrates to second population and reproduce there. So they transfer their genes to the gene pool of second population. After migration, population 2 now has a mix of gene pool. That is, it consists of the migrants and original residents. Now let's assume the migrants provide proportion M of the population 2 carrying genes from the population 1, and the original residents are represented by 1 minus M. So mathematically, after migration, the frequency of allele in the merged second population, so that is new frequency of Q in second population, equals to frequency of Q in the first population multiplied by the number of individuals which are coming from first population plus frequency of Q in second population multiplied by the resident population of 2 gives us the total change or the new frequency of Q in the second population. So you can see here that migration has changed the allelic frequency because some individual have come from another population to this population. Besides the original residents which are living here, the new entrants carries their genes here. So the frequency automatically changes. The change in the allelic frequency due to a migration, that is delta Q, will be equal to the new frequency of allele A, that is Q dash in the second population, minus the original frequency of allele, that is Q2. So delta Q will be Q dash 2 minus the original frequency, that is Q2. As we saw in the previous slide, Q dash 2 is given by Q1M plus Q2 1 minus M. So we replace this value of Q dash 2 in this equation. It comes out to be like this. If we simplify it, it comes out to be delta Q equals to Q1M plus Q2 minus Q2M minus Q2. So nullifying this plus Q2 and minus Q2, delta Q comes out to be Q1M minus Q2M, or delta Q is given by M Q1 minus Q2. So this is the overall mathematical expression where you can see the change in allelic frequencies, or you can calculate the change in allelic frequencies because of migration. It causes change in allelic frequency of population by introducing alleles from the other population. That is the common effect of migration. The magnitude of change due to migration depends on both the extent of migration and the difference in allelic frequencies between the source and the recipient population. Migration decreases the genetic differences between the population because it mixes the population, and it increases the genetic variations within the population because new migrants or new entrants are coming in the population. So this is the effect of migration on genetic difference and the genetic variation. Moving ahead to see what is the effect of genetic drift. as an evolutionary force to change allelic frequency of a population to upset the Hardy-Weinberg equilibrium. Hardy-Weinberg law requires random mating in infinitely large population to operate. But there is no real population which is infinitely large, as we have seen in the previous slides. And when population is large, when the population size is limited, the gametes that unite to form individuals of the next generation carry a sample of alleles present in the parental gene pool. Just by chance, the composition of this sample may deviate from that of the parental gene pool, and this deviation may cause allelic frequencies to change. And this effect depends on the size of the population, because the smaller the gametic sample, the greater the chance that its composition will deviate from that of the entire gene pool. Such deviations from an expected ratio due to a limited sample size is referred to as sampling error. If a population is small, a limited number of gametes unite to produce the individuals of the next generation. Chance influences which alleles are present in this limited sample, and in this way, the sampling error may lead to changes in allelic frequencies, and this is something which we call as genetic drift. So in terms of population size, genetic drift is basically a sampling error. Now, what is the cause of such genetic drift? As we just mentioned in the previous slide, the first reason is continuous small size of the population, because this small size can give rise to larger chance events, larger differences, larger deviations. So the sampling error is much more if the population size is small. The other two reasons or the cause of genetic drift are founder's effect and the bottleneck effect. Now let's try to understand these two effects and how do they cause genetic drift. Founder effect is simply establishment of a population by a new founders. Now let us try to focus on this image, which depicts the founder effects in a very illustrative way. Let's assume this is a parent population, and some of the individuals from this parent population, they migrate from here to some other place and they establish a new population here. So there is an establishment of population by some of the members from the previously existing population, and these members are called as founders. Now these population after a couple of generation changes. So founder population after a few more generation changes because it comprises of the alleles or the genotypes which are there in the founder population. So as compared to parental population, here there is a genetic drift because of the establishment of the new population by only a few founders. Similar is the effect which is called as bottleneck effect, which in other words is population reduction. Now as the name suggests, bottleneck. So you can see this example here. This is the original population. Now assume it in a shape of bottle. When you transfer some material from bottle, it passes through its neck. Now this is called as bottleneck effect. Now this term you might have heard whenever there is a traffic jam on the roads and people say there is some bottleneck because there is a narrowing of the path because of which the jam starts operating there. Now the individuals which have gone from here to here through this bottleneck, this is the surviving population, and then this surviving population reproduce and increase its numbers to a new population. So this new population is basically arising from these small surviving individuals which have come through this bottleneck effect. So this is not representing just like original population, but it solely depends on the new individuals or which have come through the bottleneck effect, and same is the case with the founder effect. So these two causes the genetic drift along with the small population size. Now suppose there are large number of separate populations, each with n individuals and p and q allelic frequencies. After one generation of random mating, genetic drift expressed in terms of the variance in allelic frequencies among the population, that is sp2, will be pq divided by 2n. The two parameters determine the amount of change resulting from the genetic drift, that is the variance in allelic frequencies. The allelic frequencies, which are P and Q, and the population size. So these are the two factors which determine the amount of change here. The genetic drift will be maximum when P and Q are equal, that is 0.5 each, and when the population size is very small. So this is the effect of genetic drift. It changes the allelic frequencies within the population. On the genetic variation, it decreases the variation through fixation of alleles within the population, and it increases the genetic divergence between the population. So these are the effects of genetic drift in total, that besides changing the gene frequencies, it decreases the genetic variation because it fixes the alleles in the population, and it increases the genetic divergence between the two populations. Now lastly, let's move to final evolutionary force, that is natural selection, which can change the allelic frequency of population or which can upset the Hardy-Weinberg equilibrium. Natural selection is the differential reproduction of genotypes in its simplest terms, and it takes place when individuals with adaptive traits produce more offsprings. Now there is a mathematical measurement tool for this natural selection, which is defined as the fitness, which is being represented by capital W. It is the reproductive success of a genotype compared with other genotypes in the population, or simply the relative reproductive success of a genotype, and its value ranges from 0 to 1. A related variable is there which is called as selection coefficient, or S, which is the relative intensity of selection against a genotype, and the selection coefficient is equal to 1 minus fitness, that is 1 minus W. So just remember these two terms in which we can define the natural selection mathematically, that is the fitness represented by W and the selection coefficient represented by S given by 1 minus W. Genetic load. The loss in average fitness of individuals in a population because the population carries deleterious alleles or genotypes is defined as the genetic load. As we just talked about the fitness of individuals and the population, so if a population carries deleterious alleles or genotypes in it, there is a loss in average fitness of individuals in that population, and this loss is termed as the genetic load. It is the reduction in mean fitness of a population relative to a population composed entirely of individuals having optimal genotype. So this term is very important when we discuss fitness of a population. Now let us understand this with an example. Let's say there are three genotypes A1, 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 A2 and A2, A2, and the mean number of offsprings that they have is 10, 5 and 2. So let us try to understand what do we mean by the fitness and selection coefficient in that. Fitness is given by the mean offsprings divided by the maximum offsprings produced by a particular genotype. So 10 divided by 10, it's 1 for this particular genotype. For genotype A1 and A2, it is 5 divided by 10, that is 0.5. For A2, it is 2 divided by 10, that is 0.2. So this is the fitness. This is the most fit. This is less fit as compared to this one, and this is the least fit one. And in terms of selection coefficient, this is 1 minus W. So the selection pressure is nil for this one. This is 0.5 for this, and this is 0.8. So that is a maximum selection against this, and that is why, because there is a least fitness in that. So here you can appreciate the link, or the relationship between the genotypes, fitness and selection coefficient based on the mean number of offsprings that they produce, that is the reproductive fitness. Now there is a method of calculating all these frequencies. For example, let's assume that the initial genotypic frequencies of these three genotypes is represented as p square, 2pq and q square, and their fitness is being represented as W11, W12, that is these two alleles, and W22. Now, the proportionate contribution of genotype to population will be the frequencies, that is the genotypic frequency of this particular genotype, multiplied by its fitness, gives an expression of proportional contribution genotype to a population. For this particular genotype, its frequency, that is 2pq, multiplied by the fitness, that is W12, and for this particular genotype, it's q square multiplied by W22. Now you can calculate further the relative genotypic frequencies after selection. So the contribution of genotypes to a population divided by the mean of fitness in each case gives us the expression for relative genotypic frequencies. Now this, what do we mean by this mean fitness, is the sum of all the contribution of genotypes to the population. So that is p square W11 plus 2pq W12 plus q square W22. Then further you can calculate allelic frequencies after selection, which can be given by P dash here represent the new allelic frequencies, that is of A1 allele is given by frequency of A1 plus half the frequency of A1 A2. So once you have these genotypic frequencies, new genotypic frequencies from this method, you can calculate the allelic frequencies. Once you have the allele frequency of P, that is the new frequency, you can calculate the frequency of Q, that is 1 minus P. As you know, the sum of the two allelic frequencies are always equal to 1. Now you can calculate change in allelic frequencies with different type of selection. Depending on the type of selection, there is a fitness value, and you can use this formula to calculate change in the Q. Now let's say selection against a recessive trait. If the type of selection is such that there is a selection against a recessive trait, the fitness value for the dominant genotype is 1. Fitness for heterozygote is also 1, but for the recessive homozygotes it's 1 minus S. So you can use this formula to calculate the change in frequency or Q. Same is the case with the selection against a dominant trait. If the selection is against a trait with no dominance, and if selection is against, that is the over dominance kind of situation is there. So you can accordingly use different formula to predict the change in allelic frequencies depending on the type of selection that is being operated. Further, you can calculate the change in allelic frequencies with different type of natural selection. Here you can see 6 types of natural selections showing the fitness relationship, the forms of selection, whether it is directional selection against recessive allele, directional selection against dominant allele, directional selection against incompletely dominant, and so on. And you can predict the results. In case of directional selection against recessive allele A2, A1 increases and A2 decreases. Directional selection against dominant allele A1, A2 will increase and A1 will decrease. So these are the predicted results depending on the type of selection, which in turn depends on the fitness relationship. Result of selection depending upon relative fitness of genotype. The rate at which allele frequency changes due to selection depends on the intensity of selection and the dominance relationship between the genotypes. Under directional selection, the rate of increase of alleles follows the relationship like dominant alleles, followed by incomplete dominant alleles, and then followed by recessive alleles. So these are the changes, rate of increase of alleles in different kinds of selection processes. Now with this brief idea about various evolutionary force in a quantitative way, let's quickly summarize what we did in this lecture. We started with population genetics, what population genetics is. Then we discussed the genetic structure of population in terms of the frequencies of genotypes and the frequencies of alleles in terms of allelic frequencies and genotypic frequencies. Then we moved ahead to the law or equilibrium or the theorem given by Hardy and Weinberg, which is popularly known as Hardy-Weinberg law. We discussed about its statement, how it says that what is the role of reproduction in changing allelic frequencies or the evolution in a population if certain assumptions are met or not. And these assumptions were with respect to the size of the population, that is, it has to be large population. It was with respect to the mating within the population, that means it has to be randomly mating. Then it was with respect to no evolutionary force, that means there is no natural selection, there is no mutation, migration, and genetic drift. And if such assumptions are met, the change in allelic frequencies or genotypic frequencies does not take in the population, or in other words, there is no evolution in such population which are following these assumptions. In other words, we can say reproduction alone cannot lead to evolution or change in the frequencies. So we discussed mathematical expression for that, and we discussed all these implications. Then we talked about individually all of these evolutionary forces, and mathematically we derived expressions how they change, how these evolutionary forces change the allelic frequencies and how we can utilize them to calculate the allelic frequencies changes in a population under specific evolutionary force, has it been natural selection, genetic drift, migration, or mutation. So with this, I would like to end here with the suggested readings. Besides the books on evolution by Ridley, by Hall and Hall-Grimson, and Douglas J. Fatoyama, I would suggest you to go through with the genetics book. Particularly, I am referring here with the Benjamin A. Pierce, that is Genetics, a Conceptual Approach. Many of the images have been taken in this presentation with due permission from the Macmillan publishers and Benjamin Pierce. So I would suggest you to go through with this population genetics chapter in this book to have a better understanding about how the quantitative studies are being carried out in terms of Hardy-Weinberg law on forces of evolution. So with this, I would like to thank you